My group and I did our authentic research on the gut microbiome. What is a gut microbiome? The collection of microbes or microorganisms composed of bacteria, bacteriophage, fungi, protozoa, and viruses present inside the human gut. The number of human genes is 25,000, but the number of genes inside a gut microbiome is 3.3 million for humans. We have more bacteria cells in our body than human cells. It is composed of symbiotic, commensal, and pathogenic bacteria. The gut microbiome's impact on health. It has an impact on obesity, autism, celiac disease, allergies, eating disorders, depression, bowel syndrome, and colon cancer. The gut microbiome and autism. Evidence that intestinal microbes exacerbate or perhaps even cause some of autism's symptoms. Research suggests that 9 out of 10 individuals with the condition suffer from gastrointestinal problems such as inflammatory bowel disease and leaky gut. A study in 2013 by Italian researchers reported that compared with healthy kids, those with autism had altered levels of several intestinal bacterial species, including fewer Bifidobacterium, a group known to promote good intestinal health. Another study in 2013 by researchers at the California Institute of Technology incited autism-like symptoms in mice using an established paradigm that involved infecting their mothers with a virus-like molecule during pregnancy. It was found that after birth, the mice had altered gut bacteria compared with healthy mice. By treating the sick rodents with a health-promoting bacterium called Bacteroides fragilis, the researchers were able to alleviate some of the behavioral symptoms. Causation and correlative. The causation was treatment with probiotic symptoms, and they were alleviated, and the correlative was found differences in gut microbiome. The gut microbiome and depression. According to a new studies by researchers at McMaster University, Canada, gut bacteria may play a crucial role in inducing anxiety and depression. Researchers subjected mice to early life stress by separating the newborn pups from their mothers from day 3 to 21 for 3 hours every day. It was confirmed that the conventional mice with complex microbiota displayed anxiety and depression-like behavior as well as high levels of stress hormone testosterone. The correlative was treatment with probiotics could be useful in preventing or improving the consequences of early life stress. Our original question was, is there a difference in gut microbiome of a healthy horse and of a demi-foal horse? Our hypothesis was both horses will have differences in their gut microbiomes. Our rationale was, according to UC Davies researchers, there is a possible link between autism in humans and neonatal maladjustment syndrome, aka demi-foal syndrome, in horses. We are going off the theory that the gut microbiome and autism are related. Based on the research, the gut microbiome of autistic individuals had 90% more Firmicutes bacteroidetes than that of the normal. We believe our demi-foal horse will have more Firmicutes bacteroidetes or another type of bacteria than the healthy horse has. Our new question was, could results be significantly different between two executions of the same fecal sample? And will the gut microbiome be different if tested by different groups? Our new hypothesis is the gut microbiome will not differ greatly. Our rationale was, according to the database, the samples were taken from the same horse, which was fed by a specific diet of whatever it was. As such, differences will not be grand, but any can be attributed to contamination. Our materials and methods are the animals we investigated. Their similarities were male horses, they're both horses, and they're between 6 months and 14 years. The differences is one was a demi-foal and one was a normal horse. Their diets were different and the exercise levels were different. There was 4 horses we compared, but our main 2 horses were the demi-foal, which is community affair, and then we also tested Pistol, which is sample number 442. Pistol is a male. He's 1 year old. He's healthy. He eats sweet food, and he has no exercise. Community Affair was a male, 6 months. He's a dummy foal. No diet was listed, and he had no exercise. The other two horses that we used in our samples was Horse, which is a male. He's 14 years old. He's healthy. He eats Purina Ultimum, and he has intensive exercise. And then Shadow, number 430. He's a male, 14 years old, healthy. He eats Timothy Hatgrass, and he has regular exercise. All but one horse ended up working. So our only horse that worked was number 361, the male that was 14 years old. He's healthy, he eats Purina Ultimum, and he has intensive exercise. Our introduction protocol. Week 1, we isolated bacterial DNA from animal scat samples. Week 2, we did quantification and PCR amplification of isolated DNA. Week 3, we did gel electrophoresis and PCR cleanup. And week 4, we did PCR cleanup 2, quantification with a fluorometer. The 3 main steps were isolate bacterial DNA from fecal matter. 2 is amplify a portion of the bacterial DNA. And 3 is sequence DNA for identification.